Ready.
Okay, so in these videos, I'm going to show you how we go about cell seeding the 6 well plates, and in the process, we're also going to be touching on passaging and cell counting throughout. So, since it's part of the process. So, first thing you want to do when you need to seed some 6 well plates is collagen coat your 6 well plates. So I have my pack here that I sprayed down before putting in here. I'm just gonna open it up, take a single plate out. Usually you would probably have to do more, but since this is just demonstration, I'll just be doing one. So kind of like close things off a little bit. And just leave it sealed. You want to try to keep it sterile on the inside. But anyways, so you have your 6 well plates. It separates just like that. It's kind of loose, so like when you're handling it, try to be careful. So what you want to do to collagen coat is the SOP goes over how you can make a collagen working solution. You just mix the collagen solution from the syringe with a 0.02 and acetic acid. But yeah, so I have this prepared ahead of time. So I'm going to set up my serological pipette. It's going to be 1.5 mils per well. So I'm going to set things up. Okay, that's kind of like where I personally want to be able to count from. So I'm just going to take this. I'm going to go up to the 0 mark, aka like 10 mils full. And I'm just going to like use the markings to count for me. Make sure you use your one-handed technique. Set this here, and yeah, just add 1.5. So I'm going to go to the 1.5 mark, the 3 mark, the 4.5 mark, the 6 mark. I overshot that one a little bit, but yeah. The 7.5 mark, and then it's going to be the 9 mark. Okay? Since I didn't touch anything, I can actually return this to the original bottle. Close that up. I won't really be needing this anymore, so I'm gonna just set that off to the side to throw that away. It might not completely cover, so just quickly do a thing like that to make sure that it's covering the whole bottom surface. So set this off to the side. And so that has to wait for an hour in the collagen solution in order for it to properly coat. So you have a couple of timers up here. I'm going to set it for an hour. Hit start. Timer's going off. And then we're going to now move on to other things. So our collagen is still waiting, and while it's still waiting, you can immediately start working on other stuff. You don't have to wait the whole hour. Or rather, you don't have to just sit there. But anyways, so I'm going to start by trypsinizing the cells. So I have my cell flask here that I took out from the incubator. So I'm just going to set them up, loosen the caps to make things easier, and then get my first pasture pipette, set it up like I showed in the other video. And yeah, you want to just aspirate the old media. So I forgot to set up a beaker in here, but fortunately this thing is right here. Just in case, I'll go ahead and re-sterilize my hands. Okay. Okay, so I've gotten rid of the old media, so now I'm good to start adding trypsin. So, what's gonna call it? I will, oops. So I have some of my trypsin here that I got from the freezer, and I also warmed up in the warm water bath along with the cell media earlier. Each glass is going to need 3 mils of that stuff. So, which essentially what the trypsin does is it like helps the cells. Well, it detaches the cells from the bottom surface when you let it sit for a couple of minutes. When you're using this stuff, make sure to not let it like the cells sit in it for too long. But I'll get more into that. So I have 3 mils here. I'm going to close the cap again. And I'm going to dispense into my flask, taking care not to touch the walls. And for now, I'm actually going to leave this upright, just because I want to wait before like letting the cells sit in the trypsin. Because if it stays in the trypsin for too long, it's bad for the cells. Since this is my last glass, it doesn't matter if I touch, so I'll just go ahead and do it. Okay. Okay, I'll throw that away in a minute. I'm going to close both of the caps, make sure it's nice and closed. Okay. Now I'm going to spread the trypsin around. To help you out, you can do a north, south, east, west motion, and just keep doing that until it coats the entire bottom. Okay, I've got it. So now I'm going to leave both of them in the incubator. And I'm going to set a timer for 10 minutes. That's usually as long as it needs in order to for the cells to detach from the bottom. Sometimes you have to give it just a little bit more time after checking to make sure that the cells are starting to come off of the surface. But never let it go above 15 minutes, because that starts getting to the point where you're starting to harm the cells a bit too much. And you might cause cell death, and you don't want that. So, yeah, 10-minute timer, along with my other 1-hour timer. So, yeah. Okay, so while you're waiting on the trypsin, you can spend some time getting a little bit ahead. So what I'm going to do is I'm going to start labeling my next set of flasks that I'm going to be passaging in. So just open it up, take the amount of flasks that you need. In my case, I only need 2. So close it back up, like seal, roll it up just to keep things nice and sealed. And then you can just put that off to the side for now. So what I'm just gonna do is I'm just gonna like label these flasks ahead of time. So what's gonna call it? The other flasks were at P36, passage 36. So these flasks are gonna be at passage 37. So I'm just gonna write P37. If applicable, you can write either A or B if you're keeping the flask separate. I'll just put that, sure. And then you want to specify what media it's in. In this case, it's a powder DMEM because we made it using the powder. What's gonna go? Oh, wait, sorry. I actually skipped the thing. Write Keiko 2. And then you can start talking about the media powder. DMEM. Plus, and then include the supplement. So in this case, it's 10% fetal bovine serum. I just abbreviate it to FBS. Plus 1% Zell Shield. Which I'll make the next one prettier because this is kind of like a bad example. But today's date is the 9th, yes?
Yep.
9/9/25, and then your initials. Okay, so that's kind of ugly, but so I'm gonna show probably what is like a prettier one. So P37B, Keiko 2, powder, DMEM plus 10% fetal bovine serum or FBS plus 1% Zell Shield, and then I can just write 9/9/25 and then my initials. Okay, so we have those labeled. So we're not going to do this today, but if you are doing a regular passaging, sometimes what you can also do is start filling up these both with 11.5 mils of DMEM. But since we're doing a cell counting to determine how much we need to add back, we're just going to leave these empty for now. And yeah, we'll just continue waiting on the trypsin. Okay, so our timer just went off. So I'm gonna, first off, I'm gonna check the cells. And so I'm gonna switch them around a bit to see if they're starting to come off. And they're starting, but it was already very confluent, so it's actually sticking pretty good. I don't know if you can quite see it, like where some of the cells are coming off, but some of them are staying. So because of that, we're actually going to just give them a little bit more time. Like I said, in other cases where like they're coming off, just go ahead and like do your stuff. But I'm just going to give them an extra 2 minutes. So yeah, we'll be back up at that. Okay, so our timer just went off again. I'm just going to hit clear, and then we'll check up on our cells again. And yeah, they progress a lot, though that can definitely be persuaded. So I'm going to get them both out. I'm going to spray them both down with ethanol before putting them in the biosafety cabinet. And so now what I'm going to do is I'm going to add 7 mils of DMEM to both flasks in order to deactivate the trypsin. So I don't know the principle behind it, but adding DMEM to trypsin makes it so that it no longer does the reaction where it like detaches the cells and like I think trypsin, what it does, I think it's like might be a protease or something, so it breaks down like particular proteins. So but when we add the DMEM, like that makes it so that the proteases like break down, I think, and so it therefore becomes deactivated. But anyways, I went up to the -2 mark for 7 mils. So I'm just going to dispense, taking care not to touch any other surfaces. Okay. And then let's get another 7 mils. And deactivate this one too. Okay, and the trypsin's been deactivated. And we'll go over what's next soon.
Okay.
Okay, so now we have our trypsinized media. So what we're gonna do now is we're gonna collect this stuff and kind of like mix it up, collect it, and then move it into one 50 ml tube. Make sure the 50 ml tube is used, is like a one that's specifically rated as sterile on the inside. So that's like, for example, this white cap ones are sterile ones. And then also I would go ahead and like label it, at least in one place, like something like original media, so that you can keep track of what tubes you're using for what. So, I'm going to take a serological pipette, and so I'm going to start with one of my flasks, and so I'm going to like essentially do a thing where I aspirate or draw it and then like dispense over and over again, and you specifically want to dispense against the wall. The reason being is there was actually some cells, you might be able to see it on the other one. I'll show you in a bit, but yeah, so I actually already got rid of most of the cells that were on the wall, but I'm just drawing it up and down. Putting it against the wall also helps to break up clumps too. So you just want to do it a couple of times. You don't want to do it like a ton, because you don't want to hurt the cells too much, and like this is kind of like a not the most fun process for them. But yeah, this is actually going to be my last dispense. I'm going to go ahead and draw as much of the media up as possible. Okay. Okay, like use better technique next time. I'll probably show you like better with this next one. So yeah, you have your media. Go ahead and dispense it into your collection tube. Okay, and then since we're going to share it in anyways, and I haven't really like touch the other external contaminated surface, I'm actually going to go ahead and reuse this one. So drop, and as you can see, there's actually some remnants of cells like on this back wall here, or maybe you can see it, I'm not sure, but there is. So I'm going to like dispense against it, and it kind of like washes them off, so that you basically now get them into solution with everyone else, which is what you want. You want as many of the cells to come with you as possible. But yeah, I'm just gonna aspirate up and down. It seems as though most of the cells have already come off the wall, but let's just mix things up a little bit more, because you kind of do want like a homogeneous mix. Okay, I'm going to draw up one last time. And yeah, that basically got as much as I can get. There might be some bubbles afterwards, which is kind of sad, but it's okay. We should have enough cells to work with. Okay. So put this off to this, well, you're going to be working with this soon, but I'll go over that soon in the next video. Okay, so now we're gonna start getting our tubes ready for counting, or our counting tubes. So I have a pack of 0.6 milliliter microtubes. This is a sterile use only one that we got from the that we're borrowing from the Moran lab. So I already sprayed it down. Just open it up. Carefully get your tubes. Don't try to be digging too much in there and touching everything. And actually, I'll take that bonus one. Sure. I wanted to anyways. So. Okay, that's nice and close. Go ahead. You don't have to do like a super detailed label. I'm just gonna write the number 1, or okay. Write the number 1 on this one. And just 2 on this one. You'll see later on why I'm doing that. But anyways, so take your P20 pipette, set it to 20 microliters. And so what you're gonna do now is, so from this room, I have something called Trypan Blue. It's like this very concentrated, strong dye. And I'm just gonna I'm just gonna aliquot 2 microliters of 20 microliters of this stuff into each tube. So the thing to note about this is, so there's some like concentrated pellet at the bottom. So don't like shake this, don't agitate it, like just draw it up the way it is. There's no need to like quote unquote homogenize it. Like, yeah, don't shake it, don't agitate it in any way. So I'm going to open it up. I'm going to use the sterile pack of pipette tips. It even has a filter to help keep things sterile. So anyways, I'm going to just draw up 20 microliters. No need to like go in very far with the tip. And like I said, you actually do want to avoid the pellet at the bottom. Take one of your tubes. So something I recommend when you're dispensing, so actually just put your tip deep in there, so that it just collects at the bottom. The reason I say to do that is because, if it's like stuck on the side of the walls or something like that, like it's a pain to try to get down, and you want it down at the bottom so that you can mix it with your media eventually. So just go ahead and like stick your tip deep in there to get it at the bottom. Just in case, I'm going to switch pipette tips before using the Trypan Blue again. Oh, and then just like you normally would, double check that you're still at the setting. I was basically still at the setting, so that's great. Still at 20 microliters. Okay, I'm going to once again stick my pipette tip in, not too far. Okay. And once again, just go ahead and stick it pretty deep in there so that it's just nice at the bottom. And yeah, okay. So we have our Trypan Blue in there. Now we need to get our collected cells in there. So before you draw up, every time make sure just gently mix, so that the cells are nice and evenly spread throughout the thing. You want that so that when you're getting your cell counting, you get like an accurate count. So I'm just going to get a new tip. I'm going to mix this around a bit more. And yeah, just stick in. Try not to touch the side of the walls with the pipette, just to avoid contamination. Okay, I have my 20 microliters. And then same thing with technique. I just like get it close to the bottom. I personally like to basically just stick it into the Trypan Blue, or rather right above it, so that it like mixes nicely. Okay. And actually, I'm gonna try to get that little dot back in too. There's a little blue dot that I'm gonna. Okay, cool. You want to avoid that. Okay, I'm going to get another pipette tip. I've double checked that it's at the 20 setting. It's great. Once again, mix around. Make sure it's a homogeneous thing. The cells try to settle at the bottom pretty quickly, so if you let it settle, you're not going to get an accurate picture of like how concentrated it is. So okay, that can be left off to the side for now. I'm going to do it with my next tube. And yeah, I'm going to go ahead and close both. And we'll go over the next steps in the next video. Okay, so this next portion, we're going to be outside of the biosafety cabinet. Yay! But anyways, so I have my 2 tubes with my Trypan Blue and my my cells. So what I'm going to want from this drawer right here, I'm going to want the hemocytometer slides. Be careful when you pick this up, like the cap comes off kind of like easily. But anyways, I'll take this one at the top. Put this back, because why not?
I'm just gonna get the name.
Yeah, Countess 2 hemocytometer. And yeah, that's the little slides. But
Okay.
yeah. So what's gonna call it? So first thing you're gonna do is you're gonna open this up. So you have 2 pieces. This part that is kind of like the foundation, and then you also have like a slide on this back. This slide is pretty fragile. Be like careful when you're working with it. But anyways, so the first thing you're gonna want to do is clean these guys up of like dust and grime. It's a little bit like grimy, I guess. So what you're gonna want to do for that is move. Okay. So make sure that it's specifically 70% ethanol you're using. One time, not while using this, but on another occasion, I accidentally used the bleach. Don't do that. Don't do that. Take your 70% ethanol over their sink. Spray, like just spray that thing. Just like mist it. So I'm just gonna shake off excess 70% ethanol a bit. So now I'm gonna dry it off using the lens paper. The way Mike described this to me, basically it's essentially an even better like Kim wipe, like even more lint free and like scratch free than Kim wipes are. So yeah, take this, start drying it off. So this the first one is always going to be like too wet to fully like dry it off. So then I throw that away. I take a second one. And once again, I just try to dry things off. The most important part is these clear areas right here, this one and this one. That's like the most important one. I'm gonna air a little bit, which by the way, make sure you have a strong grip if you do this. Like, don't throw it around the room. But anyways, So I'm going to look closely at this and see if I've gotten rid of dust particles. There seems to be a few, so I'm just going to gently wipe away. And yeah, it's actually looking pretty clear. So I'm going to leave this on this paper towel I set up for now. Next up, I'm going to do the same thing with the little slide that goes on top of it. Take it out from this little bag. Like I said, this thing is like, I mean, it seems delicate. I've never tested how delicate it is. That would be bad. But yeah, just do the same thing where you really missed it. Shake some excess off. Be gentle. Okay. And then same thing. Take your lens paper. Your first piece is gonna probably be too wet to like fully dry it. So that's okay. And like I said, once again, I'm gonna say be gentle with this. I think they're expensive. I don't know the actual price, but they give me expensive vibes. There's still a little bit of excess left, so I'm gonna get a second one to get the remaining. Okay, and then I'm gonna once again look closely and check for dust. There's a little bit more, so I'm gonna do like just a gentle, like white. And that's looking pretty great. Okay, so now what I'm gonna do is I'm gonna put this on top of these middle two ones. Try to center it, but like if it's not like perfectly centered, like it should be fine. So I'm gonna leave it like that. I'm gonna turn on the Countess on. I personally like that name just because it like not only does it like is the cell counter, but also Countess makes it sound like royalty or aristocracy. It's, I don't know, I feel like it's good marketing. Okay, so it's turned on. So what you want to do is, so I have my same pipette from earlier. If at any point you put it back in there, make sure you make sure you spray it down again. Any object that goes into the biosafety cabinet, you spray down with 70% ethanol. Anyways, so I'm going to set this actually to 12 microliters. These are non-sterile 10 microliter ones. These are okay to use outside, and actually you should use them outside of the biosafety cabinet. Okay, so I have it set to 12 microliters. I'm going to do that. And then, I'm going to take my first tube, one. So, first thing you want to do is just take the pipette tip, draw up and down several times. So, and the reason you're doing this is to like make sure it's nice and homogeneous so that you get like a nice accurate like picture of the cell concentration. We don't want the cells settled at the bottom and to get an inaccurate picture / count. So I'm just going to do that a little bit. Okay, I'm going to take that last draw, and I'm going to set the first side of the slide. So if you can see, there's actually a bit of like a permanently like stained blue area here. It's like this little ridge. So the first couple of times that I tried to like do this, I like struggled with it. So fortunately I did come up with a couple of tricks. So what I do is I just place the pipette tip like just right in there, just let it rest. I use my second hands for stability, and then you just shoot it in, and it loads via capillary action. So you see, the first time I did it, I was like, oh, I'm gonna do it like all slowly and whatnot, but you don't need to do that. Just have faith in the capillary action. Just shoot it, and it'll work out. Okay. So I'm actually going to take from that same tube another 12 microliters. I'm going to double check my setting. Okay. Take another pipette tip. Okay, I'm going to do the same thing where I pipette up and down to mix things up. Okay, that should be nice and homogeneous. Okay, and I'm going to do the same thing on the B side. Okay, great. They're nice and loaded. So now we can load it into the Countess. So this thing slides out. You can take it. I'm going to go ahead and do the A side first. So just stick it in like that with A on the left. The one on the left is the one that's going to read first. So push it in. It kind of like pushes in like a video game cartridge. It's honestly pretty satisfying. But anyways, you're going to let it wait, like give it a second to focus. Okay, it's focused. So now you can push the capture button. Okay, great. We got our first count. So, in your lab notebook, go ahead and record like these values. What's I'm gonna call it? So here, so 1A is the tube, and then the side. I'm going to put my first value 1.91 times 10 to the 6 per milliliter. That's how many cells per milliliter it's getting from this. So now you can push back on the back button, the results. So now pick a set of cells at the top for you to use as your landmark. So in this case, let's just say I'm going to use like that big one with the two small ones there. And so what you want to do is you want to slide this out so that that's just barely out of view of the screen. As you can see, that same landmark is right here, so I'm going to like get it just out of screen. And yeah, so I'm going to once again capture. Okay, 2.28 times 10 to the 6 per milliliter. Great. Once again back. I'm going to pick this like little faint one as my landmark. You can pick like more visible landmarks. I personally like I can pay attention to it and do it. But yeah, it's right there. And then it's out. Once again, hit capture. Okay. 2.52 times 10 to the 6 per milliliter. Okay, great. So, as a little note, I will mention, so make sure that when you have the bright field that you're not capturing any of the boundary. So like if I slide it more, that's the boundary of the slide right there. Don't include that. Like make sure it's the whole thing, because otherwise it's gonna get probably gonna get like an inaccurate counting, because it'll be like, oh, there's no one there. And yeah, no, so don't do that. I'm going to hit the back button on capture, and now I'm actually going to do the B side, oh, sometimes it doesn't quite catch the first time. You can just push it in again, and then it'll do it. Okay, great. Oh, and I'm gonna go ahead and put 1B. Okay, capture. Okay. 1.99 times 10 to the 6th per mil. Great. Oh, just as a quick side note, so fortunately the Countess, when it's doing its math, it already does in fact take into account that you kind of like diluted it with a tri-pen blue. It already does that. So the number you get, you can use it in your math. You don't have to do anything else special with it or like double it or anything like that. But anyways, so I had my landmark out of the picture. Let's hit capture. Okay, great. 2.39 times 10 to the 6th per milliliter. By the way, make sure you do in fact include that power. Sometimes if you have like a really low cell count, it'll be to the power of 5. So just be careful of that. Okay. I'm going to see. Okay, yeah, I'll just go ahead and do that one. Let's hit capture. Okay, that is, oh wow, a lot of percent dead for whatever reason. But anyways. Okay, so we can go ahead and unload it. And so I have my first set of counts. That one is a bit of that last one was a bit of an outlier. I'm kind of weirded out by that one. But regardless, now what you can do is take the top part off. Oh, and by the way, you can, like the original one you use, you can, what's it gonna call it? Throw it away, my one too. So, I'm gonna take this, and I'm once again gonna clean it off. We're gonna go through the whole process. I'm gonna spray it until like most of the blue liquid is off. I might have mentioned this earlier. There's kind of like just like a permanent like light blue staining on those ridges. So that can stay, but make sure you get rid of like the obvious like blue liquid. But yeah, then after you like clean these off again, you actually want to repeat the process with your second tube, which we will go over in a bit, but you just repeat the same process. So yeah, we can pause. Okay, so our 1 hour timer went off. So when that goes off, if you're not at a time sensitive step, you can go ahead and just start. You can just go ahead and do the this step. So we have collagen in here. What you want to do is go ahead and aspirate. So I'm going to take this one Pasteur pipette. It should be okay for you to use the same Pasteur pipette for all the wells in the plate. By the way, so for the I'm going to show you a bit of a technique thing. For this particular step, it doesn't matter too much. But essentially, when you're aspirating, you want to slide it down from the bottom. I don't know if you can see the little indent at the bottom or like that strip, but you can just follow that while you're aspirating. I just turned the vacuum off. You can follow it and go close to the bottom. That way you aspirate the liquid without actually touching the bottom. The reason you want to do that when you're doing, or the reason I'm showing you this is when you have actual cells in here, you could end up scraping it if you touch the bottom where the cells are. So to try to do that, do this kind of like technique where, yeah, you like follow and slide down. In this particular case, maybe you scrape a little bit of the collagen off if you don't do that, if you touch the bottom. I'm not sure. But yeah, just go ahead and aspirate the collagen solution. So even though we aspirate the collagen solution, it should still be coated at the bottom. But then you can take your Hank's Balance salt solution. Make sure this is prepared ahead of time, or you have enough ahead of time. And you're gonna add 2 mils of this stuff per well. So I'm gonna take my 10 milliliter serological pipette. I'm gonna do it to the -2 because I'm just gonna go ahead and take 12 at a time so that I can give. Oh, okay. Note, this is hard to do one-handed, so usually you want to loosen before you start trying to do one-handed stuff. So it's like this wonderful magenta color, very pretty. And yeah, just add 2 mils per well. I'm gonna go to the 0 mark for this first one. Okay, the 2 mark. Okay, 4 mark. Okay, 6. Great. 8. And then I can just do the rest for this last one. Okay, and then now you can just let it sit there like that indefinitely until it's time for you to start adding cells. But we'll get back to that later in one of the next videos. Okay, so I just finished doing a cell count, doing cell counts using the second tube that we made, the number 2 tube. So I have all my recordings. I can throw this away too. Now it's time to go ahead and clean this up and put it away. So I'm going to take off the top one again, leave it off to the side. Excuse me, France. And yeah, make sure you essentially like wash off the blue Trypan liquid and media mix off with 70% ethanol. Go ahead and get some excess ethanol off. And yeah, so for this part, you just want to mostly make sure that it's dry. Like next time when you're using this again to cell count, then you can worry about the dust. But this is just getting put away, so just dry it off. Just dry it off. You can go ahead and try to keep dust free, but yeah, just make sure it's dry. That's the main thing. Okay, great. It's pretty dry. I'm just gonna put that there. Okay. Next up, the smaller like little slide thing. Yeah, just get the spray / wash off the blue. This one shouldn't have any blue staining, so yeah, just completely get rid of any blue. Okay, now we can finish the process with the second one. Okay, that's nice and dry, and it honestly does look pretty dust-free too. So that's a great addition. So yeah, just put it back in its little bag. It's only open on one side, so you kind of just slide it open like that. And you can just slide it in, leave it on top, close, put it back in here, and then you can put your lens paper back in too. Okay, so for this next part, you just gotta do a little bit of math. It's nothing hard, so if you don't like math, don't worry. It's easy. Nice thing is, also for this part, you can go ahead and just take your stuff off for just a brief second, because you're not handling anything. So I like this part at least for giving me a break from all that. As you can see, like there's a lot of sweat and moisture that builds up. So anyways, so now what you just want to do, you just want to get your average for all of these values. So you can just get your phone out, your phone calculator. So as you saw earlier, I did get like a bit of an outlier with one of the counts, and if you come across that when you're doing it, this is actually probably my first time getting such an outlier like that. Just bring it up to Mike. I'm just gonna go ahead and include it here because this one's just for practice/demonstration. But if this was like a for real thing that we're using for experiments, definitely bring it up. Even if it's okay, I did just want to like emphasize that when something feels off to you, make sure you mention it to Mike or bring it up to Mike. It's better for you to sound a false alarm than to ignore what's a real alarm. So yeah, if something feels off to you, go ahead and bring it up to Mike. So I'm just gonna go ahead and add everyone together. You know, that's how you get the mean. 2.52 plus, and since it's all 10 to the 6, I can just use those first 3 digits to make things easier. .40 plus 2.73. Okay, great. I can sum them up. And then there's 12 numbers, so just going to divide that by 12. And we get 2.4666, whatever the heck. What's it call it? So that means we have an average of 2.466 times 10 to the 6 cells per milliliter. So we can, I'm gonna go ahead and, well, we'll leave it like that for now. And yeah, I'll go over what you do next in the next video. Okay, so this is the heavier math portion. So something to note, probably it might be a little confusing to describe with just this one walkthrough, but also the SOP that I put together also has an example of the math so that it can help you like understand. And if you also need clarifications, also of course just consult the example and maybe Mike or someone else if you need extra help. But anyways, so for this part, you are going to use your average cell count that you got. So, which by the way, so this average cell count number that I got, 2.466 times 10 to the 6, I took the average of all 12 of these numbers. That's what I did. So I just add all of these numbers up and then divide by 12 in order to get the mean of all the values, and that's the mean I got. So we have essentially 2.466 times 10 to the 6 cells per milliliter. So now you need two key quantities that you need. So I need 6 wells of cells because I just have one plate that I'm doing. So do your math assuming that you need like 1 extra well. So just go ahead and do the math. So pretending that you need 7 wells. The reason you do that, and let's just say you need 40, do 41. The reason you do that is just so they have a little bit of extra like room to work with when you're preparing your new media, your new cell media concentration. But regardless, so so you need two values to begin with. So first off, you need your total media that you're gonna need. So it's going to be 2.5 milliliters of media per well. So 2.5 times 7, that's 17.5. So you need 17.5 milliliters of total new media, or rather diluted media, let's call it that. And then, so you also need 475,000 cells per well. So the total amount of cells you need in your diluted tube is going to be 475,000 times 7, and then you get 3,325,000 cells. So how you get the amount you need, so just take your total amount of cells and divide it by your average to get the amount of media you need from your original. If you need a like brief demonstration, so let's just say I do the dimensional analysis. I'm just going to show you the units just to make things a little bit more clear. So yeah, do your little, I don't have enough room, let's not do that. So cells, and you're dividing that by 2.4666 times 10 to the 6 cells, and then on the top you have 1 mil. So basically when you do that math, you cross out the cells thing, you're left with only milliliters. So when you divide this by this, you get 1.348. I did that calculation earlier just on my phone calculator. So yeah, that's the amount of original cell media you need to mix with fresh media to get your new like cell diluted cell concentration. And that new diluted solution is what your actual cell solution is, what you're actually going to use to seed the plates. Okay. Just for demonstration purposes, you usually do want to try to be as precise as that, but just for the sake of simplicity, I'm going to do round it up to 1.35 milliliters of the original, which when I'm talking about the original, I'm talking about the thing that you trypsinized, deactivated, and extracted from and collected in that one tube earlier. That's why I had us label it. But anyways, so we need the original there. We need 17.5 milliliters total. So you need to mix that with a certain amount of, you need to mix this with a certain amount of new fresh DMEM that right from the bottle. So to get that amount, just take this, subtract by that, and you get the amount of new fresh DMEM that you need to mix with it in order to get the solution, the cell solution that you get, that you actually have to use to seed. So just to basically make it clearer, you're mixing your original cell mix that you got from earlier steps with this amount of fresh media. That's what you're mixing. in like a new tube. And then this new like solution is what you're gonna use to actually seed or add to your weld plates. I hope that was at least somewhat clear. Like I said, there's a math example at the end of the SOP to help you understand what exactly is going on. But yeah, even if this wasn't clear, you should have a chance to understand it a bit more. But we'll get into the next steps in the next video.
Mm-hmm.
Okay, so we're back in the biosafety cabinet. What we're going to do is we're going to use our original harvested cells in order to make the new solution that we're actually going to use to seed the cell plate, or the weld plate. So it's very common for there to be a lot built up down here, and actually, so don't do what I did. I kind of like let it sit for a while, and it's a little bit clumpy. Make sure you're revisiting to like mix around every once in a while, just to make sure it's nice and homogeneous. But anyways, so I'm just gonna mix around a bit. So I'm actually gonna go ahead and add my new media first, or rather like fresh DMEM that I'm mixing. So last time I calculated that I needed 16.15 milliliters of fresh media to mix with my original in order to get the desired concentration that I need to seed the wells. So what I'm going to do is, for the first step, I'm going to take a 25 milliliter serological pipette, a little bit bigger. It's not our biggest ones, the big boys we have are 50 mils. But anyways, so what I'm going to do is I'm going to bring it up to the 15 milliliter mark. So that's been graciously marked for us by the company. 15 milliliters right there. So yeah, I'm going to draw up 15 mils. Okay, I'm gonna get it nice and close to the thing. Stupid air bubble throwing off my volume. But okay, that should be pretty darn close to 15. So now remember, I said that we specifically needed 16.15. So I'm not going to stop there with the new media. So since I've added 15, I have 1.15 milliliters left to add. So I can take my 2.5 milliliter pipette. I set it to 1.15 mils. So I can just draw up that amount. And then yeah, once I add that, we should have the amount of like fresh media that we need to mix with. Okay, great. I'm going to switch pipette tips soon, or yeah. Now if you recall, I need 1.35 mils of this stuff. Yeah, that's what I need. If at any point like you can't quite remember, like go ahead and double check. There's no shame in that. Yeah, so I'm going to set it to 1.35 milliliters. Oh, I overshot a bit. Let me get back down. Okay, 1.35 milliliters. Get it nice and homogeneous so that like you're getting a good, accurate like representation of it, like you're getting it at the right concentration. Okay. add it to this one. Okay, great. And then you can mix this around. I'm going to try to get that last bit back out. Come on. There we go. Just as a side note about this pipette, it has, so instead of having the traditional like oh, push down to get rid of the tip, this one actually has like almost like 3 stops. So first stop, second stop, and then you push down even harder to get to third stop and then do that. Just wanted to mention that real quick. But anyways, so now I have my new media. This is what I'm actually going to use to seed. So what I will now do is aspirate the Hank's Balanced salt. You want to do this like right before you're about to seed. And actually, hang on just a second while I get an extra serological pipette, an extra 10 ml one. Yeah, this is what I'm going to use to get the media around. So in case I wasn't clear last time, so when I was aspirating the stuff, when I was aspirating the liquid, essentially what I was doing was I was touching the side of the wall and getting really close, like the side wall, and getting really close to the bottom without actually touching the bottom. So I was in fact touching the wall, but not the bottom, if that makes sense. So once again, gonna aspirate. So yeah, I avoid touching the bottom. Okay. Now the Hank's Balanced salt is gone. Now we can start adding our cells. So it's going to be 2.5 milliliters per well. So I'm probably going to draw up 10 at a time. So one thing to note is, well, I'll show you. I'm just going to go ahead and go up to the 10 mark when I'm drawing up. Make sure it's the new well, not the old one. If you use the old one, you're probably going to overseed, and you don't want that. Okay. And then yeah, 2.5 per well. So I'm going to do, so when you're dispensing, we want to try to get it like even, because we found sometimes it likes to like stay at the center. So kind of like while you're dispensing it, circle it like around a bit, like almost like you're applying like whipped cream or frosting or something on top of a cake or on top of a cupcake. So went to the 7.5 mark. Oh, such a pretty like red color. So I'm going to go to the 5 mark now. Okay. 2.5 is next. Okay. And then I'll just add the rest since there's only 2.5 mils left in there. Okay. Mix it around again. The cells might have settled to the bottom. And as you can see, there's very little in there. That's why we make sure to do the math so that we actually have a little bit extra. Because yeah, imagine if we only made like exactly 15 milliliters, we'd be cutting it pretty close. So don't drip it around like I just did. I'll probably aspirate that. So yeah, I'm gonna go down to the 2.5 mil mark. and then get the rest in here. Oh, okay. So that's usually gonna pop on its own, but what's the name call it? If you're worried, you can also pop it yourself. So, first off, I'm just gonna go ahead and get that like media off the top. So might as well. Yeah, try not to do that. I actually haven't usually done that before, but anyways, and then that like bubble there, you can actually use a new plaster pipette to pop that bubble. You see? It usually pops on its own, but I usually just like to go ahead and do it. So, anyways, this should be properly seated. If you want, you can like go ahead and double check that you have like enough cells. So put it under the microscope, and yeah, that's looking pretty good in that well. Yeah, let me see if I can focus it in such a way. Okay, so if you can, I don't know if you can tell, but there's like some cells already like adhere to the bottom. That's because of the collagen. The collagen helps it stick. It helps it seed better. Oh, and despite our efforts to make it even, it does seem as though the center is a little bit more concentrated. But yeah, no, it seems as though we got a pretty good seeding density. So it seems as though we got our math pretty good and our technique pretty good. So that's great. So I'm not gonna do it today, but also do make sure that you go ahead and like label the guys with the treatment or the cell wells and whatnot. But anyways, say you labeled it already, you can just go ahead, take this, leave it in the incubator, and then like the rest of your wells too. And then after 12 days is when you replace the DMEM in there with MEM. But I mean, make sure you feed it every 2 days until then. But yeah, that is how you seed the cell well plates. And then I'm gonna go over passaging in the next video.
Okay. All right, we're good.
Okay. So next up, I'm gonna go over passaging. So you need to so a little note, make sure you passage every single time. Your cells get to about 80 to 90% confluency as you observe under the microscope, which confluency just means coverage. So if the cells are covering like 80% of the surface on the bottom, or like 90%, then it's time to passage. If it gets beyond that, like you're kind of like past the point, but make sure you like passage. But anyways, for passaging, what you want to do is just harvest the cells like I did earlier, do a cell count, and using the cell count data, you can determine how you need to seed in the flask. But anyways, so when we want to passage, we want to get our cells to a concentration of 30,000 cells per centimeter squared. So because the flask is 75 centimeters squared as its bottom area, the total cells, what you need is just 30,000 times 75. You get 2,250,000 cells. So what you can do, so this is the average quantity of cells that we got earlier, 2.466 times 10 to the 6. So once again, you can divide this by this to get the amount of mils of the original media you need to mix with the fresh media. So when you do this, the math ends up coming out with a 0.912 milliliters of original harvested media. But just to keep things simple, I'm just gonna go ahead and do 0.9 milliliters. So I'm gonna do. So you need a total of 12 milliliters of media in each flask. So just 12 minus 0.9, and you get 11.1 milliliters of fresh DMEM that you have to add to the flasks. So, and by the way, sometimes when you're doing a regular passaging, you can also, whatchamacallit, you don't have to like collect it into one tube per se. It's nice if you're doing a cell counting. Sometimes you don't necessarily need to do a cell counting, but it is nice in general. I would say if you're using the cells for an actual experiment, go ahead and just do a cell count every time. But regardless, so I'm going to start doing that mixture. So what I'm going to start with is I'm going to start by adding the fresh DMEM to the flask that I labeled earlier. So, oh, and I keep forgetting. I should get more serological pipettes. Not the biggest deal, but make sure you remember. Yeah, just get a handful. And by the way, any ones that you don't use, you can return to the drawer. It's fine. Like if you don't open them, like it's fine to return. Okay. So fortunately, these do go to the 0.1 place. So I'm going to go ahead and do 11.1 on one of these when I'm drawing up. Loosen this first because it's hard to loosen with just one hand. Okay, so so up here is kind of like the 11 milliliter mark because up here is 10 minus 1, 11. So 11.1 right there. That's where I'm going to target. And okay, I've basically got it where I want it. It should be okay to touch the inside, not the outside, the inside of this thing because it is sterile when they pack it. But if you want to be extra precautious, yeah, you can also totally like be careful of that too. But anyways. Okay, once again, 11.1. Okay, great. So now we're gonna add our like our harvested cells to the thing. So don't use this one. Use your original that you did the math on. Oh, you see how it was? Well, I just made it go away, but maybe you saw how they collected slash cell at the bottom. Actually, you can see a little like thing right there. So go ahead and mix up. And that's looking pretty homogeneous. I'm not seeing, or I'm seeing a few pieces. And don't like shake it too vigorously. Don't like kill yourselves. Okay, that's looking relatively homogeneous. I shouldn't have let it sit. That's kind of like what happens. It gets a little stringy if you let it sit. So don't do that. Don't do that. Yeah, just regularly, even when you're not about to use it, probably just regularly mix up a little bit. Okay, so I'm going to call it there. And as I said before, you need 0.9 mils. This one actually, so I'm going to go ahead and use one of the smaller serological pipettes. There's a 2 mil one. I rarely use it, but it's nice when you're doing very small volumes. I'm just gonna go ahead and use this. So yeah, it has 0.9 measured right there, or rather, oh no, 0.9 is right here, not up there. Make sure you don't confuse the markings. Okay. Okay, I have it, the 0.9 facing me. Gonna mix it up one more time, or rather, right before I start drawing up. Okay. Then you can just add it to your first glass. I like to just move it around just to kind of like help it like be evenly spread. And then I just go ahead and like make sure the entire bottom coat is coated. Do like a north, south, east, west motion. To spread things around. Anyways, I'm going to do it for my next one too. Oh, okay, I overshot by a lot. That kind of rhymed. Okay. So yeah, I have 0.9 in there. Let's add that to this one. And once again, go ahead and do north, south, east, west, north, south, east, west, north, south, east, west. Okay. And then you can go ahead and check on the cell density under the microscope. What's my call it? So just turn this on. And that's actually looking okay. That's looking pretty good. That's looking like a pretty like nice dense seating. Let me check on this one too. Yeah, that's looking pretty decent. That's looking pretty decent. Those oh, I got a little clump of cells right there. Yeah, you want to avoid that. But yeah, these also get fed every 2 days. Once you passage, you can just leave them in the incubator too. And yeah, just leave them in there. And then after that, you can just start cleaning up, get rid of your old, like your old media, just aspirate it, save your remaining like solutions and supplies, along with your fresh supplies and stuff. And then yeah, that's about the whole process.